So we've already gone through attachment and penetration of the viral particle or viral genome into a cell. And we're going to kind of start off there and continue. And then I introduced a little bit about the synthesis in the last one, just kind of an overview. The thing is to keep in mind it's going to be slightly different for different types of virus, but in general, we're going to follow the same sorts of pattern. So what I'm doing now is, so you have to kind of pay attention for what is the example. Is it for a bacteriophage? So a specific complex virus that's infecting a prokaryotic or bacterial cell. Is it an enveloped virus that is infecting an animal cell? They'll be very similar overall, but there'll be certain very specific differences that are unique for each of them. And so keep in mind, the example I'm talking about now is going to be an enveloped virus that is infecting an animal cell, for example. So this is the envelope with spikes. So remember, the spikes are specific proteins coded for by the viral genome. And then inside we have the capsid with the DNA or genome. So now in this particular example, what we're going to do is have the viral envelope fuse to the membrane of the cell, and then the viral particle enter the cell, like this, with its genome there. Now, what will often happen is, to keep this in mind, with a bacteriophage, it's going to be a little bit different. There is no nucleus, so some of these processes can start sooner. With a eukaryotic cell, plant and animal cells, and fungal cells as well, we're going to have to get the genome of the virus into the nucleus, typically. All right, what's going to happen. So the genome itself could get in, but in many cases it's actually the viral particle that will enter the nucleus. So now the viral particle is here within the nucleus. All right, at this point, it'll usually be then the capsid will open up and we'll get the viral genome exposed. Now, there's a couple reasons for this. Not only is it because the enzymes that are involved in DNA replication and transcription are in the nucleus and not in the cytoplasm. That would be the first one. The other is that it's protecting the virus. When we think about this, if the viral genome were to enter into the cytoplasm here of the eukaryotic cell, there's typically, we don't see DNA in the cytoplasm of the cell. It could be potentially identified as foreign DNA and destroyed, okay, by nucleases. So here inside the nucleus, it's going to be more well protected. Right? So usually it doesn't get just put right into the cytoplasm. It goes into the nucleus. So now what we start to happen is the expression of the viral genes. And that's going to be related to the synthesis part. So usually what we have are early genes. Typically, some of the earliest genes for most types of virus are going to be genes. So these are viral genes, keep in mind. Genes coding for proteins to initiate viral genome replication. So if you remember, for example, in bacterial DNA replication, there was DNA A, a protein, a specific protein that bound to the origin of replication of the bacterial genome, and then the DNA polymerases can bind and the other enzymes would bind, and we can begin the process of replication. So what has to happen here is this viral genome needs to be replicated. But before it can be replicated, typically, the virus is coding for proteins that are going to bind to its own genome, the DNA in this particular case, and then trigger the replication process. So the first genes that are really expressed from the virus are going to be those involved in triggering or initiating the replication of the viral DNA. Okay. Once that begins, so typically replication is going to begin first before we're going to start to make any of the proteins that are involved in the viral structure. Okay. But before that happens, you're going to get specific proteins made that will trigger the replication process. So once replication begins, then later genes will be expressed. Something else to keep in mind that happens here. So this overall process, this is still here with these early genes. So what will occur is that the cell itself has, for these early genes to be expressed, inside the nucleus here we have RNA polymerase. So the RNA polymerase is going to make copies, not copies of the DNA, but it's going to read the information and make messenger RNA transcripts. So this will be our transcription process occurring. And so this is RNA coded for by the viral DNA. Now what's going to have to happen though is this RNA is going to have to leave the nucleus. Because protein synthesis doesn't occur inside the nucleus, it occurs out in the cytoplasm. What we'll usually see as well is something else that is important and unique. So viral messenger RNA gets processed, as messenger RNA would say, in a eukaryotic cell, so it needs a 5' cap. So the 5' cap, we said bacteria didn't have the 5' cap because they had a Shine-Dalgarno sequence for the binding of initiation proteins. But because this process is happening inside a eukaryotic cells require a 5' cap for the messenger RNA for the initiation complex to bind. So that has to happen to this viral messenger RNA. It needs the 5' cap added, and it gets modified. Now what's important about the modifications are this. You have alternative splicing occur. So hopefully you remember or studied this a little bit before, the alternative splicing. So this is where this piece of messenger RNA might be cut up a little bit. So if you remember, eukaryotic cells have introns, segments that are non-coding. So they typically get spliced out when the RNA is processed. Archaea can have introns and get spliced out, or they could be left in, and that's where the alternative part comes. You can splice out the sections or leave them in. You can splice out different sections and one time and other sections a different time. So this piece of messenger RNA can be spliced in different ways. So one piece of messenger RNA can come out here, and you get more copies of it. So this will keep happening, more and more copies of this. But the thing is, each copy can be spliced differently. So we'll just say maybe this one gets spliced like this. This one gets spliced like this. So I'll just use that as the splicing aspect of it. And they're all going to code now for proteins that are going to be made. So these might be different, in this case 3 different proteins. 3 different proteins coded for by the same piece of messenger RNA, but they end up being different because they're spliced in different ways. Right? And that's one way that the viral genome, which is going to be very small, can express a larger number of proteins. And so some of these proteins will be the initial ones that will come back then into the nucleus. So then they will have to come back in. These proteins will return. And then one of these proteins might be what binds to then the viral genome to initiate genome replication. So now we'll get more DNA being made from the viral genome. Now, some of these proteins as well will be transcription factors for the later genes. Later meaning after replication begins. And that's typically, now in this particular, in our example, it's DNA replication of the viral genome. Remember, later on we're going to get into variations of this, okay? Because not all species of virus have double-stranded DNA as their genome. Some have single-stranded DNA, some have RNA. The RNA could be single-stranded or double-stranded. So there's going to be all kinds of varieties and modifications of this later on. Again, this is for this specific example. All right. Like I said, generically the process is going to be the same, the steps and the logic of it, but we're going to potentially have different starting points, and that's going to be the difference we'll get into later. So, virus infects the cell. The viral genome enters into the nucleus of the cell, typically with the capsid intact, but then it's released into the environment of the nucleus. There we get transcription of some genes from that genome, but not all the genes. Only some of these early genes are transcribed. The later genes are kind of repressed right now. They're not being expressed, and they're going to need specific transcription factors and activators to be expressed later on. Those messenger RNAs will then code for proteins that will bind to the DNA and trigger replication of the genome. So that'll start happening now. In addition to that, you'll get some proteins that'll be transcription factors that will then code for other genes. So that's where we are now. So what are the later genes that then get expressed after these? Well, most of these we are going to involve proteins that are part of the capsid. We're going to have matrix and envelope proteins. So meaning later on, right, what's going to happen is, as more types of different genes are transcribed, come out into the cytoplasm, they get translated into proteins. Some of those will be, say, these spike proteins. That are going to get embedded into the membrane of the host cell, for example. Some of them will be proteins that are going to be involved in building the capsid structures, right? Viral capsids, like this. Now, many of them will have to be transported, again, still because we have a nucleus here, back into the nucleus for the overall assembly to take place, which is the assembly. We'll get into that after this process goes. So out in the cytoplasm, these proteins will start to be produced, the ones that are going to make the viral structure. They're going to be specific assembly proteins, which we'll talk about then later. That will be involved in putting the capsomeres together to build the capsid. They will be involved in moving the genome into individual capsids and so on. So that they will all have to be made. Usually at the very end, so instead of the later genes, last genes, the ones that are going to be expressed are going to be involved in whatever process involves the release of the virus from the cell. And so that could be a variety of different types of proteins depending on how it is released. So if it's a lytic virus, which means it's going to ultimately lyse the cell membrane, then there'll be enzymes released at the end that will destroy the membranes of the cell and release the viral particles. If it is not lytic, if it's lysogenic, then there'll be proteins that involve the facilitation of the exocytosis process that will then sort of reverse what we have going on here, and then the viral assembly, when it occurs here, will eventually come out into the cytoplasm. We'll get the viral particles, the capsids with their genomes, all right, coming out. And then eventually we'll get the exocytosis process that's then going to pull these viral particles into those structures, and they'll typically already have their spike proteins, and then they'll leave like this. And we'll go into the details of this next after this. And then we get the, another copy, and usually many thousands of copies of this. In this particular case, it's an enveloped virus. So the enveloped virus, these will now be leaving the cell with pieces of the cell membrane. So overall, going through synthesis, what are we talking about? What does that mean? Synthesis of really, we have viral proteins, and then we have the viral genome. All right, so synthesis here. Viral proteins and the viral genome. That's what we're really talking about. Now, as far as the proteins go, we have some of those proteins that are going to be transcription factors, right? So they're going to be involved in then transcribing other genes later on. They'll be involved in the replication of the genome. And then some of these proteins are structural for the virus itself, and actually building the capsid structure, being involved in the formation of the envelope and the spikes and the matrix proteins that might be involved also incorporated into there. So that's kind of the idea, right? Finally, then at the very end, that there will be the trigger of the release proteins that will then get that process going, but we'll kind of get to that later. All right, so far we have covered for a viral life cycle the uniqueness of an individual virus with proteins that will allow it to attach to specific host cells and only those cells. The penetration, or how does the virus enter the cell? You could have just the genome of the virus alone entering the cytoplasm. You could have an entire enveloped virus with its envelope coming into a cell. You could have, in this particular example, it's the capsid that with the genome that's being entered into the cytoplasm, and then it has to get into the nucleus to be revealed and get the genome out so that it can be copied and the transcription can occur. So what you need to do is kind of get just an overview of the whole process, but really there's a lot going on here. So there's a lot of things you have to know previously. The whole process of DNA replication, transcription, and translation. If you're not familiar with those, this will make much less sense to you. You really have to have all those things first. And so now we're going to continue this after synthesis. We'll now go into assembly next, and then finally end with release. And then we'll look at the alternative strategies that are for viral genomes that are of different types of material, the DNA versus RNA versus double versus single-stranded, and how this will vary for each of those. That'll be kind of toward the end of this.